So I am Katie Heisert from National Jewish Health, and I am moderating today with Patricia Leonard-Pendergrass, who is at UC San Diego. I'm really thrilled to be here today. Some of you may know me. I have been a macrophage immunologist for a number of years, but I have joined the wonderful field of NTM recently, and I'm now studying NTM at National Jewish Health, because what else do you do at National Jewish Health? Jerry Nick said he was going to lead us in song this morning, the Ballad of NTM, but I told him we don't have time today, unfortunately. So I wanted to give a little bit of an introduction because we do want to have a discussion at the end, and I want to sort of give you guys some thoughts to think about while you're listening to our four speakers today. These are my disclosures. So I think if you're here and you're interested in NTM and CF, I don't need to give much background, but I just want to lay some groundwork. So not all people with CF become chronically infected with NTM, but a significant proportion do, and we think the prevalence is increasing. And there's some thought that with modulators and people living longer, this prevalence may continue to increase. Those with true pulmonary infection demonstrate more rapid lung function decline. And for those of you who've ever had to treat these, you know that NTM pulmonary infections are very difficult to treat. They require weeks to months of intensive multi-drug antibiotic treatment. And these can have a lot of side effects on our patients, and it can be a very difficult process for our patients. And even with all of those antibiotics, treatment failure is not uncommon. So a better understanding of why people with CF get infected with NTM is essential for developing novel strategies for combating these infections. And so some of the things we're going to talk about today are which host immune mechanisms are key to detecting and eradicating infection, what is the nature of host-pathogen interactions in the CF airway, and how does that evolve over time, and what are mycobacterial virulence determinants. So we're going to talk about the host and the bug and the interface of the two. So we're going to start, the first part will be the host pathogen interactions at the cellular level. What actually happens when an immune cell meets NTM in the CF airway? So Dr. Leonard-Pendergrass will start by talking about innate immune responses to NTM. And then Dr. Will DePass will talk about NTM. Is it intracellular, extracellular, or both in the cystic fibrosis lung? And then for the second half of the symposium, we're going to switch to using genome-wide sequencing to understand how diversity and NTM clinical isolates influence host pathogen interactions. Dr. Andres Floto will talk about contributions of M. abscessus in host evolution to disease progression in CF. And we will finish with Dr. Rebecca Davidson talking about treatment refractory M. abscessus, genotype and phenotype of persistent infection. So while we're listening to all these talks, as you can see, there's going to be a lot of overlap in ideas during these talks. I had some ideas that I'm going to propose, and obviously we can discuss anything that comes up at the end of the session, but these are the concepts and ideas that I've been thinking about as I've been talking to our speakers. What is virulence? What do we mean when we talk about virulence in NTM? And how does virulence relate to infection persistence? Are these things in parallel or are they in contrast to each other? Do our in vitro models of studying these organisms relate to the in vivo phenotypes we're seeing in our patients? And do we need to consider acute infection and acquisition of these organisms as a separate phenomenon for what we think about when we think about our chronic infections in our patients who are persistent despite infection? So I'll let you percolate on those talks, those various topics. And with that, we will now move to our first speaker. And so I would like to introduce you to Dr. Patricia Leonard-Pendergrass from UC San Diego, who will launch this session.
Great. Thank you so much, Dr. Heisert, for the nice introduction, and thank you all for joining us this morning for this session. I am looking forward to talking with you all about innate immunity and nontuberculous mycobacterial infections in cystic fibrosis. And my only disclosure is that I've received funding from the Cystic Fibrosis Foundation. So this is just an overview of what I'll be speaking about this morning. First, I'll start out with a little bit of background on NTM infections in cystic fibrosis. And then I'll move on to discuss NTM macrophage interactions as well as NTM neutrophil interactions, because these are the two cell types that have been best characterized in the field of NTM host pathogen interactions. During that neutrophil section, I'll share with you all some of my own data on this topic. And then at the end, I will give a brief summary of what we've discussed and pose some additional unanswered questions that we're still interested in learning more about in the field. So as this audience is well aware, nontuberculous mycobacteria are a group of mycobacterial species that are found ubiquitously in the environment. They're found most often in soil and water sources, including both environmental and household water sources. There are over 200 species of nontuberculous mycobacteria, but only a small subset of these cause human disease. In the field of CF, the most commonly isolated forms of NTM are the rapid growing group Mycobacterium abscessus complex, which contains several related subspecies, as well as the slow growing Mycobacterium avium complex, which contains several separate species. NTM infections can be problematic in a number of different patient populations. This includes those with structural lung disease, of course, those with cystic fibrosis, but this also includes individuals with COPD, primary ciliary dyskinesia, and other causes of bronchiectasis. In addition, individuals with immunodeficiency can also be at risk for NTM infection. This can include individuals with chronic granulomatous disease, severe combined immunodeficiency, HIV/AIDS, and people with defects in the IL-12 interferon gamma pathway, among other immunodeficiencies. In cystic fibrosis, there are some variants in terms of prevalence estimates that are reported, but there are published data showing that prevalence is in the 5 to 10% range in the first decade of life and increases with age, with a 20% 5-year prevalence reported in the adult CF population. And as we know from our clinical experience, these infections can cause nodular and bronchiectatic pulmonary disease, as well as fibrocavitary lung disease. The CT image you see here reflects an individual with CF with NTM infection who has both nodular and bronchiectatic changes. And in addition to the imaging findings, we can see significant decline in lung function, pulmonary symptoms, as well as significant systemic symptoms. And as Dr. Heisert touched on in her introduction, these infections can be difficult to detect and even once detected, very challenging to treat. So this schematic gives a nice overview of the what is known about kind of the life cycle of NTM infection and the innate immune response. And this is modified from a review by Johansson et al. That appeared in Nature Reviews Microbiology in 2020. So as you can see to the left, there are potential environmental and other sources of infection that can lead to infection in the susceptible host. From there, the innate immune system, of course, serves as the first line of defense against these infections. Neutrophils and K cells, macrophages, and dendritic cells all have described roles in the response to NTM infection. And there's also likely roles for non-canonical aspects of the innate immune system, including epithelial interactions, as well as the critical role played by mucociliary clearance. Once the innate immune system has this initial response, additional immune cells are recruited, and classically a granuloma would form, and IL-12 interferon gamma signaling is activated. From there, adaptive immune cells can be recruited, and the infection, under ideal circumstances, can be contained. However, in some circumstances, failure to contain this infection can occur. One important aspect that may contribute to this is transition of morphotypes from smooth to rough. As we know, NTM species can exist in several morphotypes, including the smooth and rough morphotype, and has probably been best described for Mycobacterium abscessus, and that's what's pictured here. The smooth form of M. abscessus is thought to be the environmental strain, and this strain favors sliding motility and can form biofilms. The transition from smooth to rough can occur via the loss of cell surface glycopeptidolipids, and the rough form is actually thought to occur as an in-host adaptation, leading to increased virulence and promoting formation of cords of infection that can be resistant to the host immune response. However, factors that lead to failure of the host response to contain infection are still not completely understood. Lastly, it's also important to note that the actual site of NTM pulmonary infections is an area of controversy. Classically, NTM infections have been thought of as intracellular infections contained within granulomas, but there are an increasing number of reports showing that these infections can also exist as extracellular or endobronchial infections, especially in cystic fibrosis. And so hopefully we'll be hearing a bit more about this from our next speaker. So now I will focus in on neutrophil and macrophage interactions with nontuberculous mycobacteria. And as we move through some of this data, I will call your attention to the fact that many of the studies I'll be mentioning study only one species, either M. abscessus or M. avium, and may not study both. And so it's important to keep in mind that findings for one species may or may not be applicable to the others. First, we'll start off by discussing the macrophage. This schematic reflects several important aspects of the macrophage response to NTM. Like all pathogens, NTM have pathogen-associated molecular patterns, or PAMPs, and for NTM, these include several cell surface components, including lipomannan, lipoarabinomannan, and glycopeptidolipids. These PAMPs can interact with pathogen recognition receptors on the surface of macrophages. And for macrophages, it's been shown that C-type lectin receptors, scavenge receptors, and toll-like receptors are all important in the macrophage response to NTM. When NTM binds to lectin receptors or scavenge receptors on the macrophage, it can be taken up into phagosomes, which can mature and form a phagolysosome. And this would expose the pathogen to a low pH and high concentrations of enzymes, which ideally leads to pathogen killing. However, it has been described that NTM are capable of arresting phagosome maturation, allowing the infection to persist within macrophages. Additionally, when NTM bind to toll-like receptors on macrophages, this can induce signaling through MyD88 and activate NF-kappa B, which can result in the production of inflammatory cytokines. And autophagy has also been described as an important mechanism by which macrophages can respond to and kill NTM infections. Once a macrophage has been exposed to NTM, they can present antigens on their cell surface, which can lead to activation of adaptive immunity. So from here, I wanted to highlight a couple of interesting studies that focus in a bit more mechanistic detail about the macrophage response to NTM. And so the first one I'll mention is a study by Roux et al. And this study found that the smooth and rough morphotypes of M. abscessus actually have different intracellular fates within macrophages. They found that both wild type and CFTR deficient macrophages can allow survival of both morphotypes of M. abscessus. But once they're taken into phagosomes, the two morphotypes had a bit of a different response. The scanning electron micrograph here at the top on the left demonstrates phagosomes containing rough morphotypes of M. abscessus, and generally in the study they found that there were multiple pathogens within a single phagosome for the rough morphotype. However, on the right are phagosomes containing a smooth morphotype of M. abscessus, and these were a bit distinct in that they contained typically a single bacteria within a phagosome, and those are marked by these black stars. Interestingly, this distinct morphology also correlated with more resistance of the smooth form to intraphagosomal acidification, and macrophages that had taken up the smooth form also failed to activate macrophage autophagy or apoptosis. So this is one mechanism by which at least the smooth form of M. abscessus may evade the immune response and survive within macrophages. An additional study I wanted to point out is by Bernoux et al. This study looked then at the role of CFTR in macrophage NTM interactions using a zebrafish model. And what they found was that CFTR deficient zebrafish were more susceptible to M. abscessus infection. And specifically, they found that CFTR deficient macrophages were unable to restrict intracellular growth of M. abscessus. These fluorescent images you can see here demonstrate across the top the control conditions for both the rough and smooth morphotype of M. abscessus, and then the two panels on the bottom demonstrate CFTR knockout macrophages. And what you can hopefully appreciate is for both rough and smooth, there are many more bacteria contained within the CFTR knockout macrophages. So this study indicated that CFTR does seem to be playing an important role in the macrophage response to NTM. While we could probably spend this entire session talking just about macrophages, I also wanted to spend some time discussing the neutrophil response to NTM, which is also my particular area of interest. In general, I think it's important to point out that the neutrophil responses to tuberculosis have been a lot better described than the neutrophil responses to NTM, but we do have some emerging data on this topic. And I think there are several reasons that neutrophils are important to study in the field of NTM infection. Obviously, we all know that neutrophilic inflammation is the hallmark of CF lung disease. But additionally, we know that even in other conditions, neutrophils are recruited to the sites of NTM infection, and we know that neutrophils are capable of NTM phagocytosis. There are also a number of animal models that have been published in the literature that show that neutrophil deficient mice may be more susceptible to NTM infections. So this diagram kind of covers several aspects of how neutrophils may respond to mycobacterial infections in general. The neutrophils, as we know, can respond in a couple of ways to pathogens. One is through the formation of neutrophil extracellular traps, or nets. Under these circumstances, the neutrophil can extrude chromatin. This can immobilize pathogens and expose them to proteases and oxidants, which is designed to kill the pathogen, although also runs the risk of inducing host tissue damage. In addition to net formation, neutrophils can also respond to NTM by undergoing phagocytosis. Once they have undergone phagocytosis of a mycobacteria, the phagosome can fuse with intracytoplasmic granules that contain antimicrobial peptides such as myeloperoxidase. And the MPO, once in contact with the phagosomal contents, can convert hydrogen peroxide and superoxide into hypochlorite, and this is designed to kill pathogens within the phagosome. However, there are reports of NTM being resistant to killing through this mechanism. And additionally, it's also been demonstrated that once a neutrophil has undergone phagocytosis of an NTM, they can themselves be phagocytosed by macrophages, which may be another source of persistent NTM infection within macrophages. And in general, I think it's important to note that the various aspects of this mechanistic response and how they relate exactly to nontuberculous mycobacteria is still to some extent under investigation. I did want to highlight a few important studies looking at the mechanisms of neutrophil responses to NTM. And the studies I'll share with you in the next couple of slides were done by Dr. Kent Malcolm as part of Jerry Nick's lab at National Jewish Health, where I also did my research training in NTM. And some of these studies have helped us to understand some of these mechanisms of neutrophil responses to M. abscessus. First, they have demonstrated that Mycobacterium abscessus can induce a limited pattern of neutrophil activation as compared to the typical CF pathogen, Staph aureus. This first graph here at the top shows the percent of phagocytic neutrophils at a 1 and 2 hour time point for M. abscessus compared to Staph aureus. And what you can see is that at that 2 hour time point, there is phagocytosis of M. abscessus by neutrophils, but it occurs to a lesser extent than it does for Staph aureus. In addition, studies have shown that neutrophil killing of both smooth and rough M. abscessus is dependent upon neutrophil extracellular traps. The graphs here down at the bottom look at M. abscessus killing by neutrophils for both the smooth and rough morphotype. And what you can see is that when the neutrophils are treated with DNase, which inhibits net formation, there's a significant reduction in neutrophil killing compared to the control condition. In the same study, they also identified that M. abscessus does induce reactive oxygen species production by the neutrophils. However, this reactive oxygen species production is ineffective to induce pathogen killing. As you can see here, treatment with DPI, which inhibits reactive oxygen species, did not have any effect on neutrophil killing of M. abscessus. And then interestingly, they have also identified that neutrophil killing of M. abscessus is not dependent upon autophagy. Looking specifically at CF neutrophils are capable of killing M. abscessus. As you can see here in this killing assay, both a smooth and rough morphotype of M. abscessus were killed with about 30 to 40% efficiency by CF neutrophils. Although there was some evidence that the killing mechanisms in CF neutrophils may be different from healthy donor neutrophils. And then lastly, there is also evidence that necrotic neutrophil products can actually enhance M. abscessus biofilm formation. The microscopy images shown here, on the left you can see M. abscessus conditions alone, and on the right with this enhanced biofilm formation is M. abscessus in the presence of neutrophil products. So potentially, if neutrophil products contribute to M. abscessus biofilm growth, this could help explain why M. abscessus growth seems to be enhanced in neutrophil-rich environments, such as the CF lung. So I also wanted to share with you some of my own research, and I have really focused on studying the neutrophil response to Mycobacterium avium. So all of the data I just showed you was really focused primarily on M. abscessus. And so I wanted to understand whether the neutrophil responses were similar or different between the two species and how this relates to cystic fibrosis. So what I started out by doing was doing neutrophil killing assays, looking at both healthy donor neutrophil and CF neutrophil killing against clinical isolates of Mycobacterium avium. And what I found was that when the M. avium is opsonized with healthy donor plasma, there was efficient killing activity by both healthy donor neutrophils and CF neutrophils. And that is shown in the red circles here. However, when the bacteria were first opsonized with plasma that's been heat treated to inactivate complements, killing was significantly reduced for both conditions, as you can see in the blue squares. I also found that killing in the absence of any plasma components or in the absence of neutrophils was really minimal. So from this, I concluded that both healthy donors and CF neutrophils are capable of efficient M. avium killing when the bacteria are opsonized with healthy donor plasma, and this effect does seem to be dependent upon the presence of complement. So I next wanted to try and kind of better understand the mechanism of this response. And so I did some additional killing assays looking at specific depletion of various complement factors, and that's the graph here shown on the left. The red bar is that intact healthy donor serum killing at a 60-minute time point, which you saw on the last graph. When I treated the bacteria first with a complement C3 deficient plasma, there was a significant decrease in killing, indicating that C3 did seem to be very important for the neutrophil response to M. avium. Interestingly, though, when I looked at C1q depleted serum, which would inhibit activity of the classical complement pathway, or when I looked at factor B depleted serum, which would deplete the alternative complement pathway, neither of those had a significant effect on neutrophil killing. So the effect seems very dependent upon C3, but did not seem specific to either the classical or alternative pathways. And then moving to the graph on the right, I next wanted to understand whether potentially was there a defect in the CF plasma opsonization process that might explain in some way the increased susceptibility of persons with CF to this infection. So all of the data I'd shown you up to this point was using opsonization with healthy donor plasma. So in these studies, I reversed the experiment and used CF plasma as my source of opsonins, but healthy neutrophils to look at killing. And what I found was when CF plasma was used as our source of opsonins, there was a significant reduction in killing as compared to healthy donor plasma. So we learned from this that opsonization with CF plasma or the C3 depleted plasma both resulted in significant reductions in neutrophil killing of M. avium. And then lastly, because of this finding, I wanted to then look at complement levels in CF plasma to see whether there were differences. And so we looked at levels of several different complement components in plasma from healthy donors, plasma from persons with CF without NTM infection, and persons with CF with NTM infection. And what we found was that C3 and C3b were both elevated in persons with CF who had a history of NTM infection as compared to people with CF who did not have NTM infection or healthy donors. And then finally, we performed unsupervised hierarchical clustering of these plasma complement profiles to see whether there were kind of overall trends in the complement levels. And when we did this, there was a low and a high complement cluster that were identified. Interestingly, the low complement cluster contained all of the healthy donors, all of the individuals with CF without NTM, and a few of our individuals with CF with NTM. But our high complement cluster was exclusively comprised of persons with CF with NTM. So interestingly, we found that complement profiles are altered in some people with CF with NTM. And I have lots of future experiments planned to try and further understand these mechanisms. So from here, I would like to summarize and then pose a few unanswered questions to the group. I hopefully have shown you that both macrophages and neutrophils play important roles in the host response to NTM infection, but there are still a lot of gaps in our knowledge. And we also discussed my work showing that complement opsonization we found as an important mechanism for neutrophil killing of M. avium, and that there seem to be alterations in the complement profile of some persons with CF with NTM infection. Some areas that I think are important for future study for this field in general include understanding interactions between different innate immune cell types, understanding differences in the innate immune response between different NTM species, better descriptions of NTM epithelial interactions, improving our correlation between in vitro and in vivo data, understanding more of the differences in the host responses in CF, studying the role of co-infection with other CF pathogens, and then lastly, and I think perhaps most importantly, improving our understanding of the reasons why otherwise effective host immune responses fail in some individuals. So with that, I would just like to acknowledge the team I work with at UC San Diego, including Dr. Victor Nizay, who is my research mentor, the Nizay Lab, and the CF team at UC San Diego. I'd also like to thank the group at National Jewish Health, including Dr. Jerry Nick and Dr. Ken Malcolm, which is where I did most of the research I showed you, and then my CF mentors from Colorado, Dr. Stacey Martiniano and Scott Sagel as well. And with that, I think I could take one or two quick questions before we move on to our next speaker.
Thank you, Tricia. Excellent talk. And before I launch into the one question we'll ask, I am aware that the lighting is very bright on the screen, and we don't have an AV person in the room, I don't think. So if anyone knows how to contact AV, I've been trying to contact the CF Foundation, the meeting people, because I know we have some microscopy coming up and things that could use some better lighting. So I'm just sending a plea out if anyone knows how to fix this. So, hello.
This might be it.
There you go. Ask and you shall receive. Okay, we have one question which from Sarah Parsons I'm going to save till the end because I think that's a great question for the group. Brian Dobosh asks, if classical and alternative complement don't play a role for opsonization and killing of NTM neutrophils, what lectins do you think are most important?
Yeah, that's a great question. We definitely think the lectin pathway is kind of the next area to focus on. I also have some other data that I didn't show today that also shows that IgM may be involved and seems to be very important for this response. So kind of my next steps are to try and answer that question, understand whether there might be some IgM lectin interactions that could activate kind of the lectin pathway and lead to NTM or M. avium opsonization. But as of yet, I don't know which ones are important. I hope to know that maybe for next year's meeting or sooner.
All right, great. Thank you.
Thank you. Okay. Well, I would like to introduce our next speaker, who will be Dr. William DePaz from University of Pittsburgh. He'll be talking to us about NTM and whether it's intracellular or extracellular.
Perfect. Thank you so much, and thanks to the organizers for inviting me. I'm very excited to be part of this session and be talking to all of you today. So as Tricia mentioned, the title, or what I'm going to address today is NTM intracellular, extracellular, or both in the CF lung. I have no disclosures. And then first of all, I'd like to address, and there's been some good background already presented on this, but why do we care about this problem, right? Why do we care about the intracellular state of NTM? We know, right, NTM is a growing problem. The first sort of prevalence indicators we could get are from 1984, and it was about 1.3% among people with CF. The average today is around 13, but this varies by center and sort of by patient age and so forth. But these are very difficult infections to treat, right? These require long durations of antibiotic cocktails that are sometimes very ineffective and difficult for the patients. What particular problem I want to sort of address today is that there's a disconnect oftentimes. So when we take a particular isolate from a patient, we test it in vitro. Sometimes it seems like it should be susceptible to drugs. But then when you actually give a patient these drugs, they don't respond very well. So what's happening there? Why is there this disconnect between in vitro and patient tolerance? And one thing we suspect is that there's specific behaviors that NTM is undertaking during infection that cause it to be recalcitrant to drugs, right? Cause it to be difficult to treat. And some of these factors then are what I would argue to Katie's first point. The questions are what we should consider virulence factors, right? Behaviors that bacteria take on during infection that make them more difficult for us to eradicate. And as we alluded to earlier, so some of these behaviors that I'm going to touch on today are host cell invasion on the left there, and then extracellular biofilm formation on the right. So our question then, our main thesis is which of these and to what degree is NTM taking on these behaviors during infection, and are they sort of affecting antibiotic recalcitrance, our ability to treat? First, I'm going to focus on the host cell invasion side of things. So tell you essentially a little review of what's known in the field, some of the evidence we have that NTM is invading and living within host cells during infection, particularly focusing on Mycobacterium abscessus, but touching on MAC a little bit. And then I'll take you through my lab's essentially journey to try to answer this question by microscopy and where we're at. It'll be a lot of tool building, but I promise it'll be fun. Okay, so what do we know from the field? Evidence for NTM living intracellularly during human infection. As Tricia very nicely pointed out, Mycobacterium abscessus is able to invade and live within macrophages during tissue culture in vitro, right? And this varies depending on the morphology. So again, just a very brief background, smooth colony morphologies are more or less the wild type, where rough morphologies are due to mutations that occur during infection. Generally, this decreases a specific surface component called glycopeptidolipids, or GPLs, resulting in this constitutive chorded aggregation phenotype. And the fates of these two morphologies are different in macrophages in vitro. So for smooth macrophages, and again, Tricia went over this very well, so I'm just going to touch on it briefly, but you generally get a single bacterium per phagosome. You get specific then expression in Mycobacterium of some intracellular gene expression systems, specifically this ESX4 type 7 secretion system. This blocks phagosome acidification and allows sort of exit from the phagosome and replication then in the cytoplasm. In a zebrafish model then of infection, this results in a granuloma, so this sort of chronic infection state. This is different for the rough morphology. So the rough morphology, you get a few, generally a little clump of bacterial cells taking up into a phagosome. Largely because, again, when those GPLs get knocked out of the rough morphology genotype, you sort of expose these underlying cellular membrane components that interact with TLR2 and trigger this inflammatory response, apoptosis in the macrophages, and then in the zebrafish model, again, replication is extracellular cords. So this is what I would call then external extracellular biofilms. Okay, so in summary, right, Mycobacterium abscessus can do this. It can live intracellularly for a portion of time in macrophages in vitro, dependent, again, on the morphology. So what's some other evidence? So we know, again, in animal models, in zebrafish I already showed, also in mouse, it appears as if abscessus can live intracellularly. So I showed the microscopy in zebrafish. In mouse, this is just one image, but it's just the little purple dots or purple stains are Mycobacterium abscessus, and it appears that some of these are intracellular, that close to the nucleus of a host cell. Okay, so then we know this happens in animal models. There's some interesting studies looking at liposomal amikacin. So this is essentially amikacin encased in liposomes. It can kill intracellular Mycobacterium abscessus in vitro, and in clinical trials, it seems like this improves sputum conversion. And what this suggests then is that liposomal amikacin is better at crossing the host cell boundary, right? So it can get exposed to those intracellular Mycobacterium abscessus. So since this is more effective at treating abscessus in patients, it suggests that this phenotype is happening during infection. This point number 3 is more pronounced in Mycobacterium avium, in MAC, than it is in abscessus. And in fact, all of these points, 1 and 2 and 3, are also true for MAC. So MAC can definitely invade and live within macrophages in vitro, not only in animal models, but there's some very good evidence, obviously, that MAC lives within macrophages in humans. This is an intestinal sample from a patient with HIV. And without that CD4 response, MAC is able to live and divide within macrophages a lot. It's very problematic if you have an AIDS patient who also gets MAC. And so what this is showing is just those little purple cells, again, are Mycobacterium avium living within macrophages in a human tissue section. Okay, and then the last piece of evidence I'm going to provide before we go into our own little work here is that dominant circulating clones of Mycobacterium abscessus are better. So these are presumably, right, Mycobacterium abscessus clones that are adapting to live within human hosts. They're better at macrophage invasion in vitro. This suggests, right, that this is a phenotype that's happening during infection in humans, and that it's obviously improving its survival or virulence. And this is, I'm not going to go much into this because our next speaker, Dr. Floto, is basically what I'm citing when I talk about point number 4. So I'll let him tell you more. Okay. So all this combined leads us to our hypothesis in the lab is that, right, a portion of Mycobacterium abscessus is living intracellularly in the CF, along with smooth abscessus likely demonstrating more of an intracellular lifestyle than rough abscessus. And that's sort of the hypothesis we're working off. And then to test this, it requires the tools that we've been developing. We want to be able to label and image NTM directly in human sputum samples, distinguish intracellular versus extracellular in situ in those samples, and differentiate then smooth and rough colony morphologies, right? We need to be able to do each of these things if we want to test our hypothesis. So I'll walk you through where we are in each of these. I think we have sort of a workable method for each one, but I just want to sort of go through how we're doing that. So our general approach, or the framework that we work off, is this technique called MyPact HCR. So it's a technique to directly image samples in 3D in sputum. How this works is that we get a sputum sample, we fix it immediately in formaldehyde. We embed that sample in acrylamide. This provides a structural support. We clear it optically, and that involves removing SDS, which is sort of the major culprit in light diffraction in a given tissue. So once you remove SDS, you homogenize the refractive index. You can see right through that tissue. You can image it in 3D directly. And then we digest the bacterial cell walls. We label the bacteria, and then we can image. So I just want to show you a little bit what this looks like practically. So this is a sputum sample at day 0, and then after about 13 days of clearing in SDS, it starts to become transparent. You put it in this refractive index matching solution, and it becomes a see-through, basically. So you can image in 3 dimensions. And then lastly, right, we label. We label the bacterial ribosomal RNA generally to get a species-level view of what's there. We can label host components, usually DAPI for host DNA, and we can add lectins in there if we want to look at mucus. Then we image via confocal microscopy. So applying this technique, the big problem for a lot of you who've worked with NTM, you suspect that our ability to digest that bacterial cell wall is going to be difficult, and it is. It's a hard step, and that has taken us a long time to sort of optimize. What we do is we take our samples, we heat them, and then we expose them to proteinase K and lysozyme, and then do a couple cycles of this with SDS clearing in between, and it permeabilizes that mycobacterial cell wall enough that we can see them with our probe. So this is just a sputum sample unpermeabilized, and then with permeabilization, blue here is host, is DAPI, red is an NTM specific stain. So we're seeing NTM in a human sputum sample, and our permeabilization is sufficient to do that. And what you can kind of see here, but what I'll zoom in on in the next movie, is that oftentimes what we see this is Mycobacterium abscessus clustered around these host nuclei. So there's some likely extracellular clumps here. Usually you see them in small communities, and usually you see them closely associated with that host cell. So we suspect, although there's a lot of that goes between here and our conclusion, but we expect that a lot of these are intracellular. Okay, so how do we directly test that though? And this becomes the big question, is how do you label a live host cell and demarcate that host cell boundary in cleared tissue? So a lot of this becomes technical sort of microscopy questions, and it's fun. So our best now guess, our best assay for doing this is by labeling the cytoplasm, right? We clear these tissues, we remove a lot of the lipids, so we can't really label the membrane, and even if we could, it wouldn't necessarily give us a good sense of what's live in here, right? What host cells are alive. Because even if bacteria are clustered around the nucleus, if it's a dead cell, it's not really answering our hypothesis, testing our hypothesis, right? So we've tried a couple transcripts being relatively short-lived, and if they're in the cytoplasm, they could give us a good idea of the host cell boundary. And we didn't have much luck, but what we did have luck with is labeling the host ribosome, so ribosomal RNA. So this is just a Z-stack of a sputum sample, and you get a sense of the problem, right? There's a lot of nuclei here, and which of these are actual cells, and which of these are sort of just a dead nucleus that's about to be decomposed. But if we label that host cell ribosomal RNA, that's going to be in red coming up. This is what it looks like. You see specific cells that light up and a lot of them that don't. And we're expecting now that those dead ones are not lighting up with ribosomal RNA, but the live ones are. So if we use our imaging analysis software here, we can just make a mask around each of those red globs, right? And we can go through and then we can say, okay, these are likely the boundaries of the live cells. There's a lot we have to do in vitro to sort of validate this, but that's I think, a pretty good start. So then we can label all the NTM and say which of those falls within those masks and which falls without. And that's going to be sort of our demarcation of intracellular versus extracellular. At least our beta version of doing that. This is just in another sputum sample. I'm just showing this to illustrate that it is relatively bimodal, at least from what we can see, right? There's very bright signal, and there's a lot of nuclei that don't give any signal. There's a few in-betweens, but we need, in our major sort of impetus at the lab, or a major push right now is taking this into in vitro and sort of testing how long can you have a macrophage maintain its ribosomal RNA fluorescence over time when it dies. How long does it take to decompose, right? Okay. So that's where we are with the intracellular versus extracellular, and then I want to show you briefly what we're doing to differentiate then smooth versus rough morphologies. So this is hard to do transcriptionally wise. There's no specific transcripts that can demarcate a smooth versus a rough morphology. There's multiple mutations that can knock out GPLs in different ways. Sometimes you'll still get GPL transcription, but not transport them to the surface and so forth. So ultimately what we hope to do is do this by morphology. So I'm just briefly showing you our little in vitro test model for how we mimic sputum. This is a soft agar that we can embed little bacterial aggregates in. And then on the right here is what that looks like in 3D. So this essentially mimics the three-dimensional embedded environment that you come upon in sputum, right? So we wanted to just inoculate this ABA, we call this the agar block biofilm assay, with smooth or rough morphologies and look at what the morphology is going to look like. How did this manifest in 3D, right? So if we take those smooth versus rough morphologies, what we see is that the smooth morphology manifests mostly as this spherical kind of shape, right? This is a smooth ball, a circle. If we put this rough morphology in, it manifests very much, like Tricia said, as a chord. So it's these high surface area to sort of volume structures. And this is consistent across multiple rough colony isolates, and it happens in different media. We've tried this also with SCFM in the ABA, so it's not just specific to our conditions. And then how we could go about quantifying this, or how we plan to, right, is just that, the surface area to volume ratio, and a chord that's going to be much higher than in a sphere. And this is just a graph showing that. On the top is corrected surface area to volume ratio. 1 on this scale should be a perfect sphere. And I'm just showing a depth in the ABA as a means to separate the data points, but it doesn't affect much here. And you can see there's a sort of a clear delineation at about 1.2, where we can say anything over that tends to be a rough colony of variant, and anything under that is typically a smooth. So this is our plan, is to go ahead and apply this to right sputum to try to differentiate between the two. What we still have to do is determine if there's a size variation to this, like how small of an aggregate can you get before you differentiate a smooth versus rough, but we're in the planning stages of those experiments. Okay, so for our future directions, and I've kind of touched on all these points, we obviously are going to start multiplexing sputum with our NTM stain and our human ribosomal RNA stain, just to get a sense of what's happening intracellular versus extracellular, determine how long those cultured macrophages retain stable ribosomal RNA after death, not just macrophages, but other cells that we sort of suspect are harboring NTM. Label specific NTM transcripts, specifically that ESX4 type 7 secretion system that we know are induced upon entry into the host cell, as a means to say if NTM is undergoing this programmed intracellular lifestyle in response to being phagocytosed. And then label host cell transcripts via HCR or via our fluorescent labeling device, just to try to identify them essentially. It's easy for us to label any kind of transcripts, easier than proteins. So anything that can sort of differentiate host cells from one another is useful. And I'm just going to give you a little brief interlude of what we tried here. CD68 tends to be a good transcript to label macrophages for our samples, so that's in green. UB is just red here, and that's showing general all bacteria. So what we can see is CD68, it tends to work. So it looks like it's labeling specific cells. You can kind of see it clustered around nuclei. So we think these are macrophages in the sample. So we can then multiplex any of these to say, okay, if we do CD68 plus our ribosomal RNA, we can see the host, the cytoplasmic boundary of any macrophage. put our NTM stain in there and say, okay, these are NTM within macrophages. So we're working on combining all of these into sort of a nice cohesive analysis. So in conclusion from this part of the talk, right, our hypothesis is that a proportion of abscesses is living intracellularly. We suspect smooth are going to be more. We've developed the first versions, at least, of the imaging tools to test this. And as we kind of start to plan our next big experiments, it becomes important, I think, to really define what we mean by intracellular. If you look at any given CF pathogen, you expect probably to find a proportion of live bacterial cells in live macrophages, just as part of the normal sort of phagosome, lysosome killing, essentially. So what do we mean if we mean an intracellular bug? We probably mean a higher proportion of that bug in macrophages than not. We probably mean escaping that lysosome or that phagosome, which is going to be difficult to do imaging, but we're thinking about how to answer those questions too. And I think it's really going to behoove us to then start labeling the specific transcripts that we expect abscesses to express in the macrophage. I think that's going to be another good indicator. Okay, so then what about biofilms? And this is largely where the focus of my lab is, but it's going to be a very minor part of this talk, partly because I think the answer is more apparent. But if you flip our hypothesis here, it's a proportion of abscesses living in extracellular biofilms we expect, with rough isolate abscesses more commonly demonstrating that biofilm lifestyle. And I'm going to briefly go through then the evidence for this. This won't take as long as promised. So we know, just like with the intracellular lifestyle, NTM are able to form biofilms in vitro, and this is MAC, this is abscesses. These things can form biofilms. We know they form biofilms in the zebrafish model, specifically Mycobacterium abscessus, and I showed that data before. And there's also good imaging data from thin sections from human samples showing Mycobacterium abscessus biofilms. This is in an explanted lung. These are generally kind of small clusters, and in sputum samples on the right, labeling Mycobacterium abscessus, and they form at least small clumps. This is from a COPD lung resection, and the dark purple here are NTM. So you can see in the lumen, sort of in the epithelium, relatively large biofilms. So we suspect NTM abscessus is forming biofilms during infection. We see largely the same thing in our own imaging, although there's a lot of heterogeneity. So some of the samples we see sort of this disparate single cells and cells surrounding the host nuclei. But we also on the right see, this is labeling specifically NTM, Mycobacterium abscessus specifically in red. We see extracellular relatively large biofilms. And if we zoom in on this one right here and add a couple different probes, red here is the NTM specific, green is just the general bacterial probe. You can see they overlap, and there's not much else there. So this is what we think is a monospecies Mycobacterium abscessus biofilm in CF sputum. This isn't always what we see. Interestingly, in this, we see a lot of incidences of this, is that some biofilms are not monospecies. NTM is in red again, UB is green, and NTM is sort of dispersed amongst another species in a polymicrobial biofilm. We don't know what this other species is yet. We're designing probes and applying them to this sample to try to identify this. Our best guess, based on culture data, is Pseudomonas, but we do not know that. So a big part of what we're doing is trying then to identify what this other thing is, recapitulate these biofilms in vitro and in our ABA model, and test how these interactions and how these morphologies affect things like antibiotic tolerance. Okay, so future reductions for the biofilm side of things, we obviously want to multiplex these sputum samples, determine the frequency of biofilm forming versus intracellular NTM across multiple sputum samples, and then try to correlate either phenotype with rough or smooth morphotypes. Ultimately, we want to sort of understand how these influence clinical parameters too. Do this biofilm formation versus intracellular lifestyle correlate with exacerbations or disease progression in general, I think is a very important question. Okay. So with that, I would like to thank my lab. So this is Carlos, a fantastic undergrad. Mitch and Sean are both here, and they're both giving posters. So a big facet of my lab is trying to understand environmental cues that trigger biofilm formation and dispersal. And they both have very interesting stories along those lines. So if you're interested, see their posters. And Sean has done, I should mention, a lot of the microscopy I showed in my talk. Ifra is a former technician. She left for med school, but I still have her on here because I have such a small lab. I can't afford to be losing people in my acknowledgement slide. And then I also want to thank Katie, who's up here. She's been a fantastic collaborator, really our go-to for any clinical and host site advice. And all the sputum samples that I showed today were donated by Katie. Not donated by her, but she sent them. And then the CF Foundation and the Children's Hospital of UPMC. Again, I'm at the University of Pittsburgh. So thanks, everyone.
Great. I think we have time for one question. Dr. Honda is wondering, you show live dead differentiation for host, but can live dead for NTM be done at the same time using your sputum imaging technique?
Yeah, that's a fantastic question and something we think about a lot. And I think, again, so this is all fixed tissue. The membranes are gone, so we can't do the traditional sort of live dead thing. I think our best bet for this is going to be transcripts, right? They're short-lived molecules that are in a dead cell is probably going to be gone pretty quick. That's something that we all obviously have to do in vitro to test how quickly. But those intracellular specific transcripts, I think, are going to be a sort of dual purpose signal for us. They're going to tell if MTM is expressing those specific intracellular signals and if has been alive in the recent past, I think is our best bet. Yeah, thank you.
All right, I'm sure we could answer more questions now, but we're going to go to our next speaker to save time for discussion at the end, so thank you, Will. I'm going to invite up next Dr. Andres Floto from University of Cambridge. Tee it up. And we will switch over from the cellular interactions of host and bug to really understanding at a genome level what we can learn about these species and how they're interacting with the host.
Great. Hi, everyone. I'd like to thank Katie and Patricia for the invitation to talk to you today. And I was given the title, Contributions of Abscessus Within Host Evolution to Disease Progression in CF. What I thought I'd do is briefly go through some key steps in pathobiology of abscessus to put the within host evolution in context. So I'll briefly describe what we think we understand about acquisition, then just describe global spread and the emergence of these dominant circulating clones that William mentioned. And finally, to really step back and get a kind of holistic view of evolution of abscessus. I should have said I don't think I have any disclosures to mention, so I haven't mentioned them. Okay, so let me talk about acquisition then. I think this statement is true. So all abscessus is acquired from the environment. Okay, but sometimes people put it there. And I think you can view a whole spectrum of acquisition modes that are possible and occur for abscessus. So the first of those really is indirect person-to-person spread within the hospital. And that was described in Seattle and by us, and the papers are referenced here. And also within hospital is the possibility of a point source with an infected water supply or something that can cause these outbreaks or pseudo-outbreaks of NTM and abscessus. And there's one high-profile case in the US and one in the UK about to break. And obviously they're of concern because they're potentially medical legal implications to these and also opportunities to intervene. But there's also this concept of indirect person-to-person transmission within the community, which was hypothesized to be occurring as an explanation for the phylogenetic structure of abscessus. In our paper last year, we provided some stronger support based on looking at bacterial mutational signatures to implicate people with COPD in the global transmission networks of abscessus. So there's CF to non-CF transmission chains occurring around the world. And penultimately then this idea of fecal-oral route, which people hate when I describe it as this, but the idea of abscessus being introduced into the sewage, not being cleared by the water treatment center, and then infecting the potable water, and then infecting somebody else. And then finally, there's this true environmental acquisition, right? So genetically hugely diverse, almost unique isolates that are acquired. And you can imagine that there's a spectrum then of genetic diversity as illustrated here. So I'll just really mention some of the support for hospital-based indirect transmission of abscessus, which, as I mentioned, we think is through in fomites, that's inanimate objects, and long-lived aerosols. So this was an example here I show of the description that we published in The Lancet a few years ago. I'm showing you the family tree of abscessus. An individual patient is in a different color, so that's red and green, and you can see the isolates are almost identical there. These were several examples of a cluster of transmission cases where we had the ability to go back and do social network analysis and epidemiology, which provided opportunities for transmission around the same time that we thought that the bugs emerged. Paraphyletic relationship just means that one person's genetic diversity of a bug is totally encompassed within another one. We found the development of a mutation in one patient being transmitted to another patient, and using Bayesian inference, we were able to show that the most common recent ancestor occurred at the time where there were opportunities for transmission. So this was just an example of it. And we went on really to kind of explore in more detail how fomite transmission could occur. So this is published, but describes ACF center, one patient who was chronically infected with abscessus. They developed multi-drug resistant mutations, go into a room. You can see the months along the bottom, right? So it's a long period of time. Goes into the room, comes out of the room. Then patient 2, patient 3, patient 4, who we know were not infected before because we've been screening the whole cohort, go into the room for a little bit, come out again, and then subsequently become positive for abscessus. What you can see at the top in the blue lines is conventional cleaning. And in the diamonds at the far right of that, you can see that we were able to isolate abscessus from the environment despite conventional cleaning. And it's really only when we used perchlorate and hydrogen peroxide that we were able to clear the environmental abscessus. And as we might have expected, they were identical isolates and part of one of these dominant clones. So we think that this is a clear example of how, if you're not careful, you can have an environment where you're looking after patients, which is potentially a source of infection of abscessus. And as a consequence of this and other work, we were fortunate enough to influence, with the help of the CF Trust and the UK community, changes in infection control, which have led to are associated with a reduction in these hospital-based transmission routes. And in fact, we're able to also influence the building of the new hospital that we're now in with high-frequency ventilation, which we've demonstrated can clear pathogenic bacteria from the air within the hospital. This is work that was led by Charlie Howard. And you can see there's some references there. Okay, so the global spread then. So if you look at abscessus around the world and you do the family tree, you can find evidence of really deep branches, so genetically diverse isolates affecting individual patients. But you can see these clusters which are closely linked, right? A few tens of SNPs. And each of the colors here is a different geographical location. You can see that these suggest that there are dominant clusters. And in fact, you can see in this cluster from Bacilliense, isolates from Papworth, Scotland, Birmingham, Queensland, Brompton, Denmark, UNC, Chapel Hill, and Seattle are all clustering fairly closely together. So they were geographically mixed. And in fact, we extended this last year to a much wider genomic analysis of all available isolates. And now we can confidently say that there are 7 dominant circulating clones. They emerged around a similar time, and then they've spread around the world. The important things about the dominant circulating clones, they are host-evolved, and they are human-associated. That is to say, they have a very different mutational spectra from environmental organisms or other NTM, like MAC, for example. And they cluster very closely to tuberculosis. So they have, the majority of their time must be spent within the human lung. It's clear that they emerged before the CF community was large enough. They've certainly expanded within the CF community, And they've been transmitted then through these networks of CF and non-CF individuals across the world. Okay. I'll just briefly pause and just answer this question, really, which was raised by Katie at the beginning, I think, and Patricia mentioned it as well. The question of whether CFTR may play a role in other things apart from mucociliary clearance. And I'll just show you this as a bit of a teaser, because the paper, although I've put it as bioarchive, has been delayed till the end of the month. But these are iPSCs, so these are stem cells derived from an individual with CF, where we've genetically corrected either one or both alleles of the CFTR back to wild type. And so red is the native CF iPSCs, the yellow is with one corrected, and the blue is with both corrected. And we've differentiated these into macrophages, and you can see that in these in vitro models of infection, the CF macrophages are unable to kill abscessus. As you correct one and then both alleles, you get better killing and reduced inflammation. And I think Katie may have mentioned this paper already, but together with Laurent Kremer's team, we previously showed that knocking down or knocking out CFTR had a specific macrophage phenotype in vivo, at least in fish. Okay, so evolution. So it's fair to say that transmitted isolates of M. abscessus are more virulent, and I can say that confidently because there's no definition of virulence. I'm just telling you what I think virulence is, and it changes depending on what paper you read, and in fact, I'm not even internally consistent. But I'll tell you what I mean by virulence. So there are definitely worse clinical outcomes for these dominant circulating clones. In vitro, there seems to be higher infectivity of clustered isolates. And in vivo and in vitro, there seems to be increased virulence experimentally, meaning increased bacterial burden, increased inflammation. And here I just show you one example of an unclustered and transmitted clones in SCID mice performed by Diane Ordway's group. So can we define the steps then involved in this potential pathogenic evolution of abscessus? And this was work that we published a couple of years ago now, last year in fact, where we basically broke this down into 4 steps, or maybe 3 steps for abscessus, 4 steps for mycobacteria in general. And it turns out that environmental clones are pretty much all the same at the core level. And actually, what makes big phenotypic differences for these isolates is the acquisition of genes through horizontal gene transfer. And we described one such case, which was a methyltransferase that methylated DNA, completely changed the transcriptome and hence the behavior of these cells. So this is kind of an idea of saltatory evolution. So you've got basically a single jump, in this case, a single acquisition of a gene that affects global regulation, has massively changed the behavior of the clone. And so it's very clear that the dominant circulating clones have had these events that have caused this step change in evolution. I'm going to say virulence, okay? And it's all around macrophage survival. If you look then at within host adaptation, though, we have a process of evolution called allopatry. That is to say that because of the structure of the lung, we've got independent pockets of evolutionary behaviors, a competition of subclones in multiple places in the lung occurring at the same time. It's like finches on different Galapagos Islands doing their bit. They're doing their bit on their own. And then in this case, we just show you we were able to extrapolate and monitor the frequencies of different subclones over time in this individual patient. And then you can see the emergence and spread of, in first, aminoglycoside resistant, and then macrolide resistant, and then aminoglycoside and macrolide resistant isolates over time. And in fact, at the very end, so in this case, pre-transplant, or the explanted lung, is pretty much dominated by a single multi-drug resistant clone. But if you look in more detail then, what you find is this allopatry, this geographical separation, allows for parallel evolution within the lung. And so you can see multiple adaptive mutations occurring in parallel in a single individual. And in fact, we monitored 18 individuals over many years and found the same mutations occurring in the same genes, okay? And one of those was the GPL locus, and I'm using the same slide that everyone's been using of the rough and smooth, okay? But it's important. We, as a community, we've been obsessed with this smooth to rough transition, quite rightly. But it is only one of a number of adaptive mutations with massive phenotypic impact. And in fact, if you look at the variation of behaviors of abscessus, the variant to variant mutation, or the isolate to isolate variation, is much greater than the impact of GPL presence or absence. Nevertheless, you can also look at the consensus SNP across the whole global population of CF isolates and identify an overlapping set of genes that are much more mutated than you would expect, suggesting that they are involved in adaptation. What was remarkable, though, that we can map all of these genes onto a single protein-protein interaction network that all seem to be involved in the same processes within the abscesses. So through multiple routes, multiple entry mechanisms, we see this evolution driving survival within macrophages predominantly as the key adaptive step. And these within-host adaptive processes appear to prevent the survival of abscessus in the environment. And so we see this constrained evolution. The bug wants to become more and more evolved, but if it does, it's stopped. It's stuck in the patient, can't get to the other one. So when we see the isolates that are in fact able to transmit, you see an impact of antibiotic resistance in terms of fitness in the environment. And these key adaptive mutations also seem to impair survival on fomites, as we showed experimentally in this paper. And so our hypothesis then, to put this into a kind of general mycobacterial evolutionary framework, is that the final step then is, with sufficient host susceptibility and sufficient opportunities, you would start to see direct person-to-person transmission, which would kind of get rid of this constraint of having to survive in the environment. And we think that's kind of what TB did a few thousand years ago. And we think abscessus and the kind of TB ancestor kinetii are in stage 3 at the moment. There's another way of getting at within-host evolution, and that's to look at multiple isolates that you can collect from individuals with CF, and then undergo this really detailed phenotyping in vitro and in vivo. And we termed, with whole genome sequencing, and we termed this phenogenomic analysis, which has caught on, we just made it up. So it's now called phenogenomic analysis. And we were able to look at growth and different carbon sources. We were able to look at antibiotic resistance profiles, infection and survival within macrophages, infection and survival of flies. So Lucas, who led this work, injected 20,000 Drosophila with a needle. I mean, crazy, right? But it's probably worth it. And also we're able to map on the clinical metadata, okay? And this gives us a real insight into how things are moving in kind of this multidimensional phenotypic space. And what you find is that isolates cluster into 3 groups with distinct behaviors and with distinct clinical outcomes. Let me give you an example of the other things that you can do, though, right? Because these detailed phenotypic traits, you can then map the genome sequencing data onto and ask the question, well, what's driving those traits? So this is an example of what the Drosophila infection model looks like. So the phagocytes are in green and the abscessus is in pink or purple. You can see these loose granuloma-like structures. People go, oh, that's not a granuloma. And I say, well, it's as much a granuloma as the ones you see in zebrafish, right? So without the innate immunity, you do see this, without adaptive immunity, you see this kind of initial proto-granuloma. You can perform the bacterial GWAS. So this is a Manhattan plot. So the peaks are the ones that are most significantly associated. And I'll just highlight one. This is a new potential secretion system, which we think is chucking out stuff which modulates host interaction. And in fact, this trait, so the trait in the Drosophila, you can predict the outcome in patients. So with the secretion system deleted, you get more persistent infection than when it's present. How relevant this will be clinically, well, we've got a whole lot more work to do, but it is an example of the power of these kinds of phenotypic analysis in terms of answering the question about, what is this bug likely to do to an individual? So then to summarize, I've shared with you a kind of overview of where we think acquisition is going. There's multiple modes of getting the abscessus. We think indirect person-to-person transmission is through via aerosols and fomites. This has decreased thanks to interventions. There are clear implications for infection control. I am happy to discuss those later. We see this global spread of these dominant clones, which are human adapted and expanded in both CF and non-CF populations, potentially driven by people who smoke. And happy to discuss mutational signatures. They were published in the Nature Microbiology paper this year, and we have a bioarchive if people are interested in more general bacterial mutational spectras. And so evolution then starts off with these saltational jumps in environmental clones. There's this allopatric evolution within the lung, so within host parallel evolution, and then this idea about constrained evolution because of this requirement to go into the environment before picking up another host. And that's all I was going to say. I'd like to thank Josie, Chris, Lucas, Sophie, Aaron, Karen, and you in my lab, Julian and Izzy at the Vet School and Sanger Center, our other collaborators, and our clinical partners, and our funding, particularly the CF Trust, that have been funding this work, and we're very generously. Thank you for your attention.
Thank you for a great talk, and the work is really remarkable in the terms of the scope of what you've been able to do. I actually wanted to ask you a question based on your beautiful Nature Micro paper that you were putting up slides about. So you do find the 3 clusters. I wanted to see if you could speak to the way the clusters aligned with the patient phenotypes. And it seemed that the ones that had the most profound in vitro virulence, killed the macrophages, killed the Drosophila, were the most likely to persist in humans. And you touch on this idea that are they evolving to be more immunoevasive? Can you speak to that?
Yeah, so I think that's a great question. And it comes back to this, what is virulence, right? So is virulence my ability as a bug to cause disease? And the answer is probably that's what we mean. But actually what we look at is infection, replication, inflammation. Right. And we see 2 things. We see adaptive evolution to prevent uptake and improve survival within macrophages, right? And actually that by itself is sufficient to get more productive evolution because you've evaded, as you say, the immune system to the point at which you can then cause huge amounts of tissue damage and problems. But if you are a very virulent in the molecular biology sense, right. In the biochemical sense, not biochemical, in the microbiology sense, that is to say you burst open the macrophage, right. Then you get gobbled up.
Right.
Yeah. So it's a very good question. Yeah.
All right. We'll have more questions at the end. Thank you very much.
Thank you. All right. Well, I would like to next welcome to the stage Dr. Rebecca Davidson, coming to us from National Jewish Health, to discuss with us treatment-refractory Mycobacterium abscessus infections.
So first of all, I just want to thank Katie and Tricia for inviting me to speak here in this really wonderful session. I love to see how all of the stories are weaving together and all the progress that's being made. So I'm going to talk today about the genotype and phenotype of persistent infection with a little bit of background info to start. So I have no disclosures. So first I'd like to start just by highlighting some of the CFF-funded research projects that are going on at National Jewish Health. So really kind of amid the concerns about the potential for person-to-person transmission of NTM, those early reports that Andres touched on, the CF Foundation funded the Colorado NTM Research and Development Program back in 2015. And the purpose of this was to analyze whole genome sequences of NTM isolates sent from US CF centers, so really all across the country, sent to National Jewish, so that we could identify the frequency of genetically similar clusters within US CF centers. And then in 2020, we transitioned to the Colorado National Resource Centers, where we basically were using that same infrastructure to provide clinical trial support in terms of performing whole genome sequencing, providing clinical NTM isolates for compound testing, and also development of culture-independent markers for detection of NTM during infection and to monitor treatment response. And so through these programs, we have supported multi-site clinical trials, including the PREDICT, which is the PREDICT trial, which is a prospective evaluation of NTM disease in CF, and the PATIENTS trial, which is the prospective algorithm for treatment of NTM and CF. And the purpose of these two trials is really to, these are efforts to develop and validate standardized protocols for the diagnosis and treatment of pulmonary NTM. We also support the HALT NTM trial, which is the healthcare-associated links in transmission of NTM. And this one is a systematic evidence-based approach to investigating potential episodes of healthcare-associated NTM transmission within a single care center. So this trial was actually a follow-up to our original RDP project to really follow up on these genetic clusters that we identified within single centers. So really the thing that, all of these programs have in common, and what we really relied on was the willingness of all of the CF centers in the US to get your isolates, mail them to us so that we could do whole genome sequencing. And so really, through this process, we've been able to sequence over 1,000 NTM isolates from the US. So first of all, we just want to say thank you to everyone who helped us out and participated in this. And we'd just like to share what we have been able to learn by analyzing the sequences that you have provided to us. So the first study I will just talk a little bit about is our study of population genomics of M. abscessus from US CF centers. And so this is just showing a phylogenetic tree of isolates from M. abscessus subspecies abscessus, and you can see our sampling geography in this map up above. This is looking at 204 isolates, one isolate per patient. And if we look at the phylogenetic relationships, we see the two dominant clones, part of the ones that, or a couple of them that Andreas described in his talk, so DCC1 and DCC2, and these make up over 50% of the isolates in this study population, while the rest of them are more genetically diverse. Now, if we look at the same type of analysis for M. avium, which is a slightly smaller sample size but similar geography, similar geographic sampling, we see that for avium, there really aren't any dominant circulating clones. They're much more genetically diverse, and if I showed you the tree of M. intracellulare, it would look basically the same. So the other thing that we've learned through this analysis is that not only are M. abscessus infections clonal within a patient population, or have some clones in the patient population, the infections within a person are also clonal over time. And we were able to observe this looking at longitudinal isolates from 108 subjects from this study. And so the graph on the left here is just showing you the duration of time between the first and last isolate from each subject. So some of the duration was anywhere from a month or a couple months to 3 and 4 years. And so if we look at the distribution of pairwise genetic distances, and when we compare their genomes of within patient isolate pairs, that's what we're showing in the histogram here. What we can see, it's kind of hard to see here with the with these colors, but the darker blue is the within patient distribution, and at most in the isolates that we looked at, the SNP distances within patient were only 30 SNPs in their core genome, which is pretty darn similar compared to the between patient population, which has a broader distribution. Now again, this is really not the case for MAC. It's pretty much a different story over there, wherein a similar analysis in our MAC population, out of the 55 subjects that had 2 or more isolates, so around a third of them had multiple diverse strains that were very genetically diverse, and this is very likely an underestimation. So I think just looking at this information, and it's been mentioned multiple times today, all NTM are not created equal, not all NTM infections are equal, and this also may give us a clue about where people may be picking up these infections and also the risk for reinfection over time. So I just want to touch on this HALT study just a little bit, just to show you other ways that we have used our genomic data to analyze these genetically similar clusters. So now we're going to zoom out from the US and we're going to zoom into just a single center investigation at our Colorado site. And here is showing a phylogenetic tree that was published, and this paper was published led by Dr. Jane Gross back in earlier this year. It's showing the phylogenetic tree of the M. abscessus subspecies abscessus from the isolates collected from this center. And what you can see again is the clustered isolates, dominant circulating clone 1, and then the colored circles are actually the genetic clusters that were originally identified as having a SNP threshold of less than 30 SNPs. This is between patient. But since we already knew they were clustered, and Jane will talk about some, in her talk later today, she'll talk more details about the HALT study, but For this study, we added an extra layer of genomic comparisons, also comparing the accessory genomes between these clustered isolates to look for really a more higher stringency level of relatedness between isolate pairs, so that which may indicate more recent acquisition from a common source. So on the x-axis here, we have the pairwise distances for all the isolates in the cluster. So you can see the SNPs only go up to 30, so they're very similar by that sense. But now the y-axis, we're looking at their percent shared genome, or the percent of the accessory genome that they share. And in this box up here in the upper left corner, this is sort of our high level of stringency where they are highly similar at both the core and the accessory genome levels, which I think gives us higher confidence about whether they may have been acquired from the same place. And so Jane will be speaking in the workshop W22 this afternoon if you guys want to hear more about HALT. Okay, so now I want to circle back to the original title of the talk and really studying the genomics and the evolution of treatment refractory M. abscessus infection. And really we got the opportunity to study it in a lot of detail in this paper that this case study that we published in Cell earlier this year. And so the case here is a 26-year-old male with CF who had his first positive culture of M. abscessus in 2016, and then he was enrolled in the PREDICT trial. He met diagnostic criteria for NTM lung disease in early 2017 and then was enrolled in the patient's trial. So he underwent antibiotic treatment, but the treatment did not go away and it was refractory. And I'll show you some more details to that. So we sent the isolates to Graham Hatfield's lab at the University of Pittsburgh for phage screening, and he was able to identify 2 effective phages that were active against this isolate, and the patient was initiated on phage therapy in September of 2020. And with a combination of phage therapy and continuing the antibiotic therapy course, he achieved culture conversion in January of 2021. So this sustained culture conversion made him eligible for lung transplant, which occurred in October of 2021. And he completed phage and antibiotic treatment earlier this year and still remains culture negative. So here's just a graphic showing the clinical course of this subject. So in this graph, the y-axis is the FEV1 and the x-axis is the days since the initiation of phage treatment. So that dotted line over time 0 is the day of the initiation of phage, and then the green line, vertical line, is the time of transplant. So this is a very complete history here. I want to kind of draw your attention to the top row here, which is the NTM culture history on the top. So the red slash lines are culture positive for M. abscessus, and the black lines are culture negative for NTM. And so you can see that there was a decline in FEV1, especially around the time of the M. abscessus infection, which continues to decline over time. So here's a graphic here just showing his antibiotics over this time period. And so he rotated through several antibiotics over this time period, but at any one time he was on at least 4 or 5 drugs for potentially treating NTM. So, my interest is in bacterial genomics, so I'm going to focus on the evolution of the bacterial population. So for this part of so I was mainly involved with this part of the study where we really analyzed in a lot of detail 40 isolates collected on 36 occasions across this time course. So because he was enrolled in the PREDICT in patients trials, we had most of the isolates from the first positive culture through the last positive culture spanning 1,800 days. And the analysis metrics I'm going to show you are the ones that I've already talked about from our previous studies, looking at core genome comparisons, which are single nucleotide polymorphisms or SNPs. Then we use these SNPs to construct a time-scaled phylogeny and estimate the mutation rate. And then we can also look at the accessory genome composition to look at isolate similarity over time. And just this hypothesis is that these in-depth genomic comparisons will allow us to really look at the population dynamics and adaptations to treatment. So just high-level results. In the clinical lab, and we verified it, was classified as M. abscessus subspecies abscessus with full-length ERM genes, and they were all ref morphology. And there was no evidence of mutational resistance in the ribosomal RNA genes. The isolates were classified as dominant circulating clone 1. Yes? Oh yeah, sure. Sorry, guys. And so in the core genome, we only identified 50 SNPs across the entire data set, and there were no more than 11 SNPs distance between any two isolates across this entire almost 5-year span, which is really, I think, pretty remarkable. In the accessory genome, we did not observe any plasmids or intact prophages in these isolates. From these SNPs and the time course, we were able to estimate a mean substitution rate of the population at 2.7 times 10 to the minus 7 mutations per site per year, which is a pretty slow evolution rate, not as slow as TB, but faster than TB, but not as slow as, or slower than E. coli or Pseudomonas. And this mutation rate is very comparable to what has been described previously for DCC1 and other longitudinal isolates. So now I'm just going to walk you through our timescale phylogeny, because even though there were not a lot of SNPs that we identified, we can still really track the mutations over time in this phylogeny. So this figure shows the timescale phylogeny of isolates collected approximately 4 years before the initiation of phage through the last positive culture, observed 245 days after the addition of phage. So the tree shows the phylogenetic relationships among the isolates, which are indicated by the colored shapes at the end of the branches. The most recent common ancestors are shown as open circles at the nodes of each branch. And based on the estimated mutation rate, we estimated that the basal MRCA emerged approximately 30 months before the first positive culture. So now if we start from left to right, the pre-treatment isolates shown as black triangles, they're relatively diverse, at least they're on the top and bottom of the tree. After the initiation of antibiotic treatment, new sublineages emerged, and they're labeled here as A, B, and C, each having their own unique functional mutations, which are shown as red letters above each branch. And then after the initiation of phage, only one sublineage was observed, suggesting sort of a selective reduction of the isolate population by the phage treatment. However, we did recover one persister isolate after 3 months of negative cultures from the lower lineage, suggesting that this isolate may have evaded antibiotic and phage treatment, maybe by hiding out in a cavity or something like that. So I told you that we only saw 50 SNPs across the whole population, but only 34 of them were functional SNPs, functional mutations in 29 genes, and this would be SNPs that may cause non-synonymous or nonsense mutations in the proteins, potentially affecting protein function. So among these functional mutations, we also identified parallel evolution of adaptive mutations, although it's a different set of genes than Andrea showed us earlier, which I think is so interesting. And they both happen to be from the same pathway, which is potentially regulating metal ion transport. So this top gene in yellow here, this MNTR, was actually mutated here on the left branch, the upper branch, and it was a early termination codon, which potentially abolishes function, although we haven't validated it, in that entire upper branch. And then it was mutated again in 2 other branches on the lower lineage. So the MNTH was also mutated in 2 different branches of the tree in the lower lineage. So I think this is a very interesting observation and may potentially represent a pathway that was not necessary anymore because of different nutrient availability in its new environment. So then we also looked at the accessory genome evolution, and so this is just showing a heat map of the presence and absence of genes in these 40 isolates. So in this one now, the rows are genes and the columns are the isolates. This is not time scaled, but the isolates are in the same order as in the tree. And so blue here means the gene is present and the cream color means the gene is absent. So there were only 153 accessory genes identified across this sample set. And you can see in the pre-treatment isolates and the early isolates, there's kind of more diversity in the genes that are present or absent in this population. But then after antibiotic treatment, after about a year after antibiotic treatment, you can see how it starts really getting more narrow genetic diversity, and there's just a lot of similarity between the isolates. We also observed that there was a group of genes that appeared to be lost 1 year post-antibiotic treatment and that were not observed at any point after that, which may potentially affect some of the adaptations we could see in the airway. So really to follow up on this study, we have really to kind of understand what are these adaptations that are happening in these isolates over time. They do have some, genetic diversity, but they're also quite clonal. So what we've done is we, in collaboration with Jennifer Honda's lab at National Jewish Health, we have started looking at some in vitro phenotypes of some of these isolates. So we picked 5 isolates spanning the 4 years of infection, including 1 pretreatment isolate and 4 isolates post-antibiotic and 1 isolate post-phage. And the first question we asked is, do the isolates have altered growth phenotypes? And so this is just a graph showing the log CFUs 24 hours post-inoculation in 7H9 media. And what you can see here is that the isolate 1 corresponds to the early isolate in order. So the earliest isolate has robust growth in 7H9 media, but the adaptation over time is the bacteria grows to, at least in this in vitro system, is a more slow-growing bacteria, which it consistently decreases as the isolates were exposed to antibiotics. So the next question we asked was whether the isolates have an altered infection phenotype. So in Jen's lab, they performed in vitro infections with these isolates in healthy alveolar macrophages from 3 donors. And here is what they saw. So here is the log 10 CFUs, 1 hour post-infection with the macrophages, looking at macrophage uptake. What they saw was that the pretreatment isolate, or what you could think of as the potentially environmental isolate, had very robust uptake in the macrophages, which slowly decreased with each subsequent isolate, independent of the genetic lineage. Then they carried this experiment out and looked at macrophage survival and growth inside. And these 2 graphs here, we're just looking at the early isolate, number 1, and the latest isolate, number 5. And what we found was that the early isolate, pretreatment isolate, had very robust survival and growth in the macrophages, but the furthest isolate out that was isolated 4 years later actually did not grow in macrophages and nothing was recovered. And then we see the corresponding activation of TNF-alpha that corresponds to the growth in the macrophages. So I think this data is very preliminary, but it's very exciting. And we don't know if this is the correct phenotype to look at, but we do see a potential transition from an environmental phenotype to an airway lifestyle, which I think really dovetails with what Andres was talking about in his evolution piece. And so we don't know if this would really translate across all of subjects, but Jen and I have been funded by the foundation to evaluate some other in vitro phenotypes on a larger set of case series. So in conclusion, the M. abscessus population, both between patients, but especially within patients, is very clonal. And in our case study was very clonal over 4 years of infection. We observed adaptive mutations in the genome after sustained antibiotic treatment, including parallel evolution of genes associated with metal ion and transport. And we also observed that a subset of genes were lost approximately 1 year after antibiotic treatment. So we showed you the reduction in the accessory genome and really isolating down to that 1 lineage in the phylogeny after antibiotic and phage treatment, which really shows that in this case, the persistent infection was selection for a persistent and homogeneous population. And we actually believe that this low mutation rate and genetic stability of the isolate population was what actually probably made the phage successful and did not result in selection for antibiotic-resistant isolates, or phage-resistant isolates, rather. And our preliminary in vitro data suggests a particular type of transition, an adaptation to an airway lifestyle. So there are just tons of acknowledgements. This is mainly the acknowledgement for the phage study. I probably don't have everybody that was involved with all the data that I showed, but just want to thank everybody at National Jewish Health, our partners at Pittsburgh, at Children's Colorado. We also have collaborators at the University of Colorado School of Medicine and Colorado State, and really just thanking all of you for participating in our research and the foundation for the funding. So thanks, everyone.
So one question that I had was, it seems like for the isolates that you tested longitudinally from the phage patient, there was kind of narrowing diversity over time. So that would suggest then that it's a single time point of infection that has caused a persistent infection. But I guess I always wonder, do we think that's generally the case, or do we have genomic data to say whether there are patients actually getting repeated infections from environmental sources?
Yeah, I mean, that was kind of like what I was alluding to in the early part. So I think there are many studies from us and from the European isolate collection just showing that the M. abscessus populations are highly clonal over time, even after many years of infection, but doesn't seem to be the case for all of the MAC infections. So I think that that's something that's really distinct between the two species. Hope that answers it.
Perfect.
Thank you. We're going to ask you to stay on the stage. We're going to ask our other three presenters to come up so we can have a discussion. We've been accumulating your questions so we can ask everyone, so thank you. All right. Hopefully everyone can fit and not fall off the stage. We have some focused questions. Spencer Poore had a great question, which is near and dear to my heart. If I can change this over, I have it here. Okay, so this is a panel question. Thank you all for participating. I feel like we have these great perspectives that, as people have said, have dovetailed. So in NTM and CF, you see kind of what he's referring to as classic airways disease with proximal bronchiectasis and then distal airway findings. The CF is an airways disease. So he says, with macrophages residing in the alveoli and neutrophils being recruited to the lung, and I will add, Spencer, monocyte macrophages being recruited to the airways as well, both seem to be involved in killing. Has anyone looked at the spatial immune response within the airways, given the intra and extracellular behavior in the organism and abnormal mucus in CF that likely varies in relation to the differing epithelium from proximal and distal airways? And have we done any studies where we've looked at the genomes in particular areas of the lung? So whoever wants to start, there's a microphone. I will say, I will speak about spatial location in the lung and NTM tomorrow, this afternoon in a workshop. You can either go to Jane's workshop or my workshop, but
This is mostly a wish list, but we've been confined at least looking spatially to sputum, but at some point to sort of get at that more, I think getting explanted lung samples and sort of analyzing those as far as interactions with the epithelium and intracellular versus extracellular is something that, at least with our capacity, we're interested in doing. I don't have any insight into it right now, though.
Do you have any available samples? It's a great question. The lung is a really complex environment. I know that, like in Pseudomonas, Pradeep Singh's lab has been able to, there was several talks at this meeting about bronching in different areas. Bronchoscopy in CF is obviously a topic that we are discussing a lot right now in the absence of sputum, and it would be very cool to be able to look in different locations of the lung with different levels of damage to see what the NTM populations look like. As Andres pointed out, there was a lot of pictures of smooth and rough bacteria.
Same picture.
What do we know about the rough NTM and antibiotic resistance, and whether it's intra and extracellular? Anyone here have?
What I know, both rough and smooth are equally antibiotic resistant in terms of, at least in vitro, if you make them form biofilms, biofilm formation confers resistance. But I don't think there's any, correct me if I'm wrong, anyone, intrinsic difference between the two, rough or smooth, as far as just antibiotic resistance if you test planktonic cells or at least small clumps. That's my understanding. And then the intracellular versus extracellular is sort of the smooth, at least in the zebrafish model, tends to survive and live sort of more intrinsically intracellular, whereas the rough one breaks out quicker and lives extracellularly. And that's sort of what we expect, but please expand.
I'm going to let you into a dirty secret, that there are actually many more morphotypes than just rough and smooth. And actually, for those NTM aficionados that spend time in the lab, you'll see rapid transitions in both directions, which are not due to mutations in the GPL locus. So I think we're really scratching the surface of morphotypes, but I do agree with William that there's no bacterial intrinsic differences in antibiotic resistance.
All right, so we'll talked a little bit about other infections that happen in the CF airway. It's unusual that our patients have only NTM. And so Megan Kodrowski asks, if immune cells are infected with intracellular NTM, do you think this can affect their ability to kill or interact with other pathogens or co-infections? So what do you think is happening with Pseudomonas? Does anyone have thoughts on that?
You mean co-infection?
Co-infection. This is an example. This is such a great questions, and so much science needs to be done.
Yeah, you all have the same questions we have. So yeah, I think, I don't think there's a great, clear answer to that, but I do think there is some evidence that, if you're in the case of a Pseudomonas infection, if there is increased, neutrophil, infiltration and necrosis, that the neutrophil products may then enhance formation of biofilms and allow NTM actually to persist longer in the airways. So I think there is at least some in vitro evidence that could be one mechanism that would explain maybe why we're seeing those co-infections, but there's still a lot unknown. I don't know if others have comments.
I've got a tangential comment.
Yeah, please.
This is my tangential comment. So if you look at macrophage migration and you feed macrophages stuff that they can't digest, then they stop. They can't move. Right. So I do think that there's loads of possibilities for interactions, but I think it's a great question. Yeah.
And I think that one of the things, getting back to what is virulence and what is the mycobacteria? Is it an active process that it's changing the host cells? Is it injecting things from a secretion system? We know from other bacteria that the host cells can be hijacked by those secretion systems. And so is that changing how the macrophage or neutrophil might interact with another bacteria? We just don't know, but these are great questions. There's been a couple of questions in the list of questions about how does Trikafta affect all of this? Jerry Nick left a comment because he has data from the PREDICT and PROMISE studies about what we've been observing in terms of NTM sputum positivity. So because we don't have an audience microphone, he left a nice response. I can say that in our center, that when people are coming for NTM referral for treatment, if they haven't been diagnosed with the cause of their bronchiectasis, our infectious disease doctors will work them up, and if it turns out they have atypical CF, they will not treat the NTM until we've started people on CF treatment, particularly if they can get a modulator. And a number of our patients have improved enough that we don't treat their NTM once they've started modulator therapy. So this is just anecdote right now, at least, and then numbers of cases. What's exactly modulators doing, we don't know. And then I don't know if anyone else from the panel wants to speak to modulators. And Tricia left a comment about what she sees with neutrophils killing.
At the risk of drying up funding for this area, I think Becky Prevost published earlier this year registry analysis showing decreased levels of new acquisition of NTM. So the paper that I kind of I teased you with demonstrates a mechanism of CFTR function within macrophages, which is responsive, at least in vitro, to Trikafta effects. What does that mean? Who knows? But yeah. We have the same anecdotes, actually, about patients. Yeah.
Did you want to speak to your observations?
Yeah, and the comment I left was just in regards to neutrophil killing of MAC. When I compared CF on highly effective modulator versus off, there wasn't a difference in killing, which makes sense, I think, given that the neutrophil killing of the CF neutrophils was similar to the healthy donor neutrophils in the first place when they're opsonized with healthy plasma, but of course they're not. So we're still doing more studies along those lines. But I think we'll still have, obviously cases of patients who are not eligible for modulator, and there's still going to be a subset that don't clear their infection. So
yeah, as a reminder that people with non-CF bronchiectasis get NTM too. So in terms of funding drying up, I think we can safely say we're still going to have an NTM problem. It is a question, I think, with Trikafta, as our patients age and their lung disease develops over time on modulators, what is the niche in the lung that NTM likes? Are we going to see increase patients with NTM over time? Will that we don't fully understand which lungs are most susceptible to NTM, and maybe as people age and develop, kind of smoldering bronchiectasis independent of CFTR dysfunction, we might actually see more NTM. That's my hypothesis. So, Some targeted questions. Dr. Pendergrass, are there any tests to assess complement function beyond complement levels? And are there any drugs that enhance complement function? And I will add to that, what do we know about complement levels in the BAL and sputum?
Those are great questions. So yes, there are assays that can test kind of complement activation, and so we've started to do a little bit of those studies, but we don't have the definitive answer yet. So I think it's a great question, because if levels are not different or in fact elevated, is this instead reflecting not a difference in the quantity but in the activity? So that's ongoing. In terms of agents to enhance complement function, of course this is something I've looked into with these findings. I haven't found a lot that would be directly activating. There are obviously complement inhibitors out there, but I think potentially an area, if this turned out to be a useful therapeutic target that might be worthwhile would be there are also a lot of kind of endogenous inhibitors of complement, so potentially inhibiting an inhibitor might be easier than activating. So I think it could potentially be amenable to therapy, but there's not an obvious existing therapeutic that would do that right now. And the last part was about complement in the airways. So there is a lot of kind of historical data, especially like from the 70s and some more recent papers that have looked. There are definitely complement factors and antibodies in the airways. There's also a lot of data that airway proteases may really inhibit the function of airway opsonins because they can kind of degrade antibodies and degrade complement to where they're less effective. So that is one future direction I'm hoping to look at, is really what's going on in the airways, and our protease is part of the problem. And there's definitely some clinical data kind of correlating complement levels with lung function as well. So I think it's definitely there in the airways, and that's my next big step, I think, for this work.
All right, let's talk a little about antibiotic resistance. Andres, this was directed to you, but I think Rebecca could probably weigh in as well. Any implications for treatment, considering that there are more morphotypes and virulence factors than we thought, and based on the whole genome sequencing you've been doing and knowing what the patients have received and what we thought was resistant, what comments about antibiotic resistance, potentially new directions we should be taking for how we treat these infections?
Yeah, I mean, I don't really know where to start, except that, I think we have some kind of go-to, genetic mutations that we have been studied quite a bit, which is like mutational resistance in ribosomal RNases, and then the ERM48 or ERM41 inducible resistance to macrolides. So, so I think that, I think the phage story really kind of showed us that there could be some other possibilities for treatment, and maybe we need things that are kind of attacking this. And I'm not a clinician, so I'm just going to kind of speak as best I can here, but I think maybe we need some things that are sort of attacking these like alternative lifestyles, whether it's, that they are living in biofilms and maybe they're transitioning to that over time or something like that, I think might be something to think about.
That's a good answer, actually. That's better than my answer. Okay, so let me break it down. So you can analyze you can analyze essentiality and vulnerability of genes in abscesses using methods similar to the ROC group have done for tuberculosis, and you can find a whole host of potential targets which haven't been accessed by therapeutic antibiotics yet. That is hope for the future. What I would say is that in terms of predicting resistance, once you move away from aminoglycosides and macrolides through ribosomal RNA mutations, it becomes really difficult. And I'll just give you one kind of nightmare scenario, which is that we induced resistance to a whole host of antibiotics in the lab, and in between each step up in concentration, we grew them without antibiotics present over weeks and weeks and months and months and months, and thought, well, we'd whole genome sequence them to see what mutations we found. And the vast majority of them had no mutations. So this was heritable resistance to antibiotics with no mutations or copy number changes. So there is something really weird going on there. So anyway, I'll leave you with that.
So, and an interesting thought here, I was thinking about this during Rebecca's talk, which is you looked at the early and late isolates and saw that the later ones were slower growing and didn't grow as well in macrophages. It would be really hard to break this down because these patients have all gotten so many antibiotics, but how much do we know about if these in-host evolution, lifestyle changes are due to the host response or to the antibiotics? Can we break that down? I mean, obviously, if you see an antibiotic resistance mutation, you can say, okay, there's that and there's less fitness. But, traditionally, smooth to rough has been described as just the host factors changing the bacteria. So thoughts on that? What do we think in terms of the in-host evolution? What's our primary driver? Are we causing it with antibiotics, or is it going to happen regardless?
Yeah, I mean, I guess from kind of the population biology point of view, you're kind of thinking about, which lineages are being selected for over time. And there are many really intense selection pressures in the airway, especially over time, right? So I don't really know what is causing what, but in those last slides I showed you, and Jen and I have talked about it a lot because I'm also not an immunologist, but I just wonder if, this reduced uptake and growth in macrophages, is that, because, the macrophages were more effective at controlling it, or is it more that the bugs were evading the macrophage response, right? So the latter makes more sense to me in it, the fact that it may be evolving in that direction.
I just want to quickly shout out my student, Sean, who has a poster, but we've seen, at least with some wild-type smooth variants of abscessus, right? If we evolve them just in SCFM, so the synthetic cystic fibrosis medium, we actually change biofilm phenotype. So our original idea was that the smooth to rough could be just due to environmental, and we're not quite seeing that, but we're definitely seeing changes in how they form biofilm. So they're more biofilm, or more robust biofilm formers just by growing in that nutritional environment. So that is at least part of at least that.
What I can comment on the morphotype stuff is Jean-Louis Herman's group have looked in mice, and you see spontaneous development of rough. So there's an example where there's no antibiotic at play. Right. The slow growth that Rebecca mentioned in the talk is interesting because generally bacteria that grow slowly are more resistant to antibiotics, right? And interestingly, certainly in E. coli and other gram-negatives, mutations in central metabolism, which slow growth, are associated with antibiotic resistance. So that may be an example of something there.
And of course, classically, our antibiotics work by disrupting replication. So the paradigm with tuberculosis is when it's latent and it's not replicating, that's why the antibiotics don't work. Right. So there's now a push to try to get TB out of latency to try to improve antibiotics in that setting. So I, of course, will put it a push here that we should be doing immunomodulatory therapy to try to get the host to kill the bacteria better, since that's my area of interest. But I think, this understanding how the bug changes, both in relation to its replicative capacity and its metabolic state, as well as its in-host interaction, is going to completely affect how we treat these organisms. And I think we need to come up with creative new methods.
Yeah, I totally agree with that. And as one of the major culprits of promoting multi-antibiotic therapies, I was really struck by Roy Kishoni's group did an amazing paper which came out a couple of weeks ago, where they looked at interactions on killing of lots of different antibiotics. And I just worry that actually we're making life harder for ourselves by combining antibiotics in the wrong way. That's really worrying, I think.
And then just as a separate point, Jody Corley, who's a postdoc in my lab, came up with this idea, which I think is really interesting, is we give our patients with CF a lot of medications and a lot of antibiotics, and we think of these antibiotics as not being mycobacterial, but is it changing the mycobacteria? Are they responding to those antibiotics even though they're not susceptible to being killed by them? What do we know about that? I don't know if anyone has any thoughts about how any of these other compounds, I don't know if this has been studied at all. The things we think of as non-traditionally antimycobacterial still could affect the metabolism and growth state of the mycobacteria. So more scary thoughts to go home with. I think I've reached all the questions in the chat here. If anyone had further questions, please, you can even just stand up and ask. We have a few more minutes.
Can you say that last part? Say that one more time.
Metal ion pathways.
Oh, yeah, no, really, I don't really have any more information beyond what I showed you.
The question.
I think the question says that were we able to kind of further study this metal ion transport pathway kind of from the host side? Is that Okay. Yeah, no, and like I said, don't really have any other information beyond what I showed you, except, kind of thinking about, just nutrient availability and, where this thing was acquired potentially from the environment versus what they see in the airway. I mean, that in an evolutionary sense, makes some kind of sense of why it may, be acquiring mutations in this pathway that it may or may not need anymore.
Well, with that, I'd like to thank all of our speakers today. Thank you very much. Thank you for coming.